I'm going to talk about DNA. When you talk about DNA, DNA stands for deoxyribonucleic acid. D is for the sugar, N is for nucleic, and A is for acid. So the first thing that we're going to talk about is we're going to talk about DNA structure. So this should be a review because we learned this when we learned biological molecules. Now, deoxyribonucleic acid is a polymer. It is what we call a polynucleotide. So when we talk about DNA structure, the first thing that you need to understand is DNA is a nucleic acid, which is a polynucleotide. The second thing is you need to understand the building blocks of DNA. The building blocks of DNA are called nucleotides. So a nucleotide is literally a monomer. It is the monomer, which means one unit of DNA. Now, when we look at the nucleotide structure, it is composed of three things. The first thing that you always talk about with the building blocks of DNA and nucleotides is it's got a 5-carbon sugar. So this is what a nucleotide looks like. Now each point of the house represents a carbon. So this is the 5-carbon sugar. Deoxyribose. We know that deoxyribose is the 5-carbon sugar of DNA because it's part of its name. Now, this is basically the centerpiece. So nucleotides have a 5-carbon sugar, deoxyribose. Now coming off the left of it, it is physically bonded to a phosphate group, which we just draw as a circle. This is what we call a phosphate group. And then to the right of our 5-carbon sugar, I'm going to give it a little space, we have nitrogen base that is attached to a 5-carbon sugar. I'm just going to write NB. This represents a nitrogen base. Now, the nitrogen base, there can be 4 of them. They can be adenine, thymine, guanine, and cytosine. So we have 4 nitrogen bases. Only one can attach here at a time, but there are 4 of them: adenine, thymine, guanine, and cytosine. So when we talk about DNA, DNA is a polynucleotide, so you're going to have repeating units of nucleotides that just are connected. Now, when we talk about DNA, these nucleotides are connected to form a long chain. That's where we get the name polynucleotide. Many of these are going to be linked together. So if we were going to look at DNA, DNA structure is a double helix. So first thing is you can start by drawing two strains that kind of twist over each other. This is a double helix, but what you don't see is that this line represents the sugar and the phosphate. So the line that is here represents deoxyribose, which is in the center. It's also on this side, deoxyribose. And going to the outside is the phosphate group. That's what the line represents. So this whole thing would be basically representing polynucleotide. So right here we have 3 on this side and 3 on this side. So when we talk about this line, when we talk about DNA, we call that line the sugar phosphate backbone. That's what the line represents. So literally it's deoxyribose, which is the sugar, and phosphates attaching to the outside. That's the line, the sugar phosphate backbone. Now, facing towards the inside are your nitrogen bases. So your nitrogen bases actually complementary pair. Every time you have an adenine, it will complementary base pair on the opposite side with thymine. And every time you have guanine, it will complementary base pair with cytosine. So the way that I remember this is at Garden City, because I went to school in Garden City. So the nitrogen base, adenine, will be here. So the opposite complementary nitrogen base would be thymine. So you go to the next sugar. If we said that the base pair here is guanine, then the opposite side would be cytosine. If this one was thymine, then the opposite side would be adenine. Now, it's easiest to just draw tick marks. The tick marks represent the nitrogen bases on the inside. Those represent your nitrogen bases. So you literally can put any letters on here. I'm just going to say C, C, C. So the opposite side would be guanine, 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 because these are always going to complementary base pair, because there are 2 strains or 2 strands. It's a double helix. So if this one is adenine, 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 then the opposite side is thymine, thymine, thymine. So it's a double helix. But when we draw it, the simple way to draw it is the sugar phosphate backbone of the nucleotide, but it is a polynucleotide. So technically this strand here is 1, 2, 3, 4, 5, 6, 7, 8, 9 nucleotides with nitrogen bases. Now, there's bonds involved. When talking about bonds, we have to physically connect our nitrogen bases. So the bond that is going to hold our complementary base pair our nitrogen bases together are called hydrogen bonds. So hydrogen bonds are going to link our nitrogen pairs together. So you literally can go through, and I'm color coding mine to make it a little easier, and draw a blue line to represent hydrogen bonds. So it's a double helix, and the nitrogen bases are going to complementary base pair with hydrogens. Now, to link together your nucleotides, we use a special bond here as well. So the line that bonds your sugars together is another bond represented. This is called a phosphodiester bond. So if you say phosphodiester bond, these are going to link nucleotides together to form DNA. The phosphodiester bond is between nucleotides, so there's 3 nucleotides here. The bond between nucleotides is called phosphodiester. Hydrogen bonds are going to link complementary base pairs. So when we draw DNA as a double helix, you now are going to say that this is called the sugar phosphate backbone. The little ticks towards the inside are the nitrogen bases, which complementary base pair by hydrogen bonds. And that's pretty much what we talk about. Now, the other thing that we talk about is we say that DNA is anti-parallel. Now, I could go into a long spill about the chain or the orientation of DNA. When we look at the sugar phosphate backbone, we can count that this is a 5-carbon sugar. So 1, 2, 3, 4, 5. Now, the phosphate and where it's oriented and where it binds is going to determine the orientation of the phosphate bound to it. Instead of going into all the phosphate groups, I just use the word that says number 5, DNA is anti-parallel. They're opposite directions. They go in opposite directions. So one strand is going to go what we call from 5', and that would be where the phosphate is attached to it, to 3'. And the other strand is going to be 3' to 5'. Don't worry so much about how the phosphate's oriented, but understand the word anti-parallel. So if you write that this is the 5', you follow it to the opposite side and you say this is the 3'. Well, they go in opposite directions. So if this is 3, this one will be 5'. Go to the opposite side and it is 3'. So they're complementary to each other in the reverse direction. And that's what anti-parallel means. It's the orientation of the nucleotides. The chain of nucleotides are oriented from 5' to 3' on one strand, and the other side is 3' to 5'.